Hi everyone, today we're going to introduce a medication that is going to change your eye color. My name is Wenxuan, and this is my team partner.
Hi everyone, my name is Sherry.
The name of this chemical is bimetaforst. It have a more common name being Lumigan. The primary use of this medication is to treat hyperpressure in your eye, and its secondary use is more closer to our life, which is enhancement in our eyelashes growth. The disease that it can cure is pretty serious because the high pressure in your eye can damage your optic nerve and cause possibly permanent blindness.
And for the chemical structure, it has 25 carbon, 35 hydrogen, 1 nitrogen, and 4 oxygen. And the 17-phenyltreonine prostaglandin F2 alpha is a metabolically stable analog for PGF2 alpha, and it's a potent agonist for the FP receptor. It binds to the FP receptor on alveolar luteal cells with a relative potency of 756% compared to the value of the PGF2 alpha. And notice the ethyl amide group. This group is very crucial for its biologically activity. And also, you can see the stereo centers, and their specific configuration is key to the drug's effectiveness.
There are multiple ways to synthesize this drug. The synthesis process we choose to share today involves four steps. The first one is a reaction called the Horner-Westmore-Edmonds reaction. So over here, this process enabled the aldehyde to turn into carbene, and in the second step, the ketone group is being reduced into alcohol. The third step started by a reduction of esters over here, and then proceed by using the Witten salts to form another double bond. In the end, the carboxylic acid is reacting with NH2 group, which will form the crucial amide group at the end of the chemical. The first step of the synthesis is initiated in the basic condition. The hydrogen on this carbon will be pulled off by the base in the solvent, and then its lone pair, while being stabilized by the two double bonded O on right next to it, will attack the carbonyl over here. In this attack, it will form a temporary intermediate where the PCCO four elements will be very close to each other and form another four-member ring intermediate over here. This is unstable, so it will quickly break into two double bond products, which is this one is a byproduct, and this one is the product that will be extracted and used in the following steps.
And in step 2, the sodium borohydride acts as a reducing agent in this reaction. First, the nucleophilic BH bond will attack the electrophilic ketone carbon and pushing the electrons up to the oxygen. Then the lone pair on the negatively charged oxygen will grab a proton from the environment and then from the OH group, thus completing the reduction of carbonyl.
And in the third steps, dibyl H is being used in the reaction. Dibyl H will help the ester to reduce, but not completely into carboxylic acid, but reduce into the ketone aldehyde form. It will start by the lone pair attacking the Al in the dibyl H, and then the negative charge over here will help pulling off the double bond, the pi bond on this oxygen into a lone pair. Then it will help push off this O group, which will then help breaking the CO bond, eventually reaching the ideal product over here while retaining its chiral center. So we can see from the starting material in this step, the stereochemistry is being preserved in both in both carbon, so that the OH group and the aldehyde group in the end still have the stoichiometry that we need. And then in the second part of the step 3, the reaction is initiated by the hydrogen on this carbon in the Witten salt being pulled off by the basic environment. While the carbon is having a lone pair, it is being stabilized by the positive charge phosphate over here, and then the lone pair will attack the carbonyl on this aldehyde. It will follow a very similar step in the step 1 of this synthesis, where the PCCO4 elements form a intermediate of a 4-member ring. Again, with the formation of 2 double bond at the very same time, the product that we need will be formed here with the byproduct of this PO double bond that is not being shown.
And for the last step, when it comes to the final step, the lone pair on nitrogen attacks the carbonyl first and forming a negatively charged oxygen, which then act as a nucleophile. And then the lone pair on oxygen attacks the hydrogen bonded to nitrogen, which then form a good leaving group H2O. And finally, the lone pair on oxygen comes down, help the H2O group to leave, and then form our final product.
In terms of the future development of this drug, it can be seen that there are a lot of chiral center in this chemical, and in some steps, because of the racemic mixture that is formed and the need to separate them via chromatography, the yields in some steps is about 30%, which is not very ideal. So scientists are figuring different ways to improve the selectivity in each step, using catalysts that can only form the targeted product without forming the other one, so that it reduces the need to further work up and purify the product.
Then the cosmetic industry is keenly interested in bimatoprost, particularly for eyelash growth enhancement. And research is underway to optimize dosage and application methods to maximize cosmetic benefits while minimizing the ocular side effects. And further, research might explore its potential for treating other hair growth disorders, offering a new horizon in dermatological treatments. And finally, research is also focusing on making bimatoprost more accessible and affordable, particularly in developing countries where glaucoma is the leading cause of blindness. Novel manufacturing processes, alternative synthesis methods, and global health initiatives are part of this effort, aiming to bring this crucial medication to a broader population.
And thank you so much for listening to our talk in this medication.